Next up, we have our second plenary on engineered T cells as novel treatment for MS, NMOSD and MOGAD, presented by Professor Hans-Peter Hartung. I cordially invite Dr. Rukshani Rupasinghe and Dr. Dulmini Veeratunga to introduce our esteemed speaker.
Thank you. We now proceed to the next plenary session. It's a pleasure to welcome back Professor Hans-Peter Hartung, who will deliver a talk titled Engineered T Cell as Novel Treatment for MS, NMOSD and MOGAD. Professor Hartung, over to you, sir.
Thank you very much, Chairs. Thank you very much for asking me to talk about this subject. Engineering of T cells allows to construct T cells carrying a receptor specific for a defined antigen or autoantigens, and this is termed chimeric antigen receptor, CAR. CAR T cell therapy has revolutionized the treatment of hematological malignancies. It's rapidly emerging as a transformative approach for treating neuroimmune diseases such as myasthenia gravis, MS, NMOSD, and MOGAD. These conditions, as you know, are driven in part by autoreactive B cells that sustain chronic inflammation and progressive tissue damage. While current immunomodulatory therapies have improved clinical outcomes, they often require lifelong administration and fail to effectively eliminate compartmentalized inflammation within the central nervous system. Recent advances in CD19 and BCMA-directed CAR T-cell therapy demonstrate the potential to achieve targeted, durable B-cell depletion and immune reprogramming in autoimmune disease. You see here a timeline describing the various milestones in CAR T-cell development, really going back into the 80s of the past century. Against this background, I'd like to discuss this kind of innovative approach as it relates to MS, NMOSD, and MOGAD. Now, I mentioned the malignancies, and there are a number of FDA and EMA-approved CAR T-cell products. And this is a listing as of, I think, March of last year. So we do have these CAR T-cell products. We know their safety profile, and therefore it's quite, in a way, comfortable to explore their utility in autoimmune diseases, in particular those that we are interested in. And you see here from Nature a News and Views article to indicate that over the past years the exploration of engineered T-cell therapy for autoimmune diseases has significantly increased. Now, we know that MS is a complex disease. B and T cell functions and antibody-mediated mechanisms play a central role in MS pathogenesis. It's important also to recognize or recall that there is a very intimate, orchestrated, bidirectional, tripartite immune cell-CNS interaction in multiple sclerosis. So you have the T cells, you have the B cells, the myeloid cells, and they act upon the central nervous system. And the pathogenesis is certainly multifaceted, but for example, T cells do play an important role, particularly in the interplay with B cells. Now, as you may remember from medical school times, the T cell receptor is important in sensing or recognizing antigen or autoantigen on antigen-presenting cells such as B cells, macrophages, dendritic cells, and microglia. And if one looks at the molecular structure, you may remember that there is a constant region and a variable region, and here with the alpha and beta chains you see the antigen receptor, the antigen recognizing part of the receptor. Now, with chimeric antigen receptor T cell engineering, you replace some part of the T cell receptor by a target element, the one that you would like to have the T cells home to recognize. And there are various ways explored initially by June to sort of insert genes with this different makeup to generate CAR T cells. So just to have a look at the procedure, you leukapheresis patients to obtain T cells, you culture the T cells, activate them, stimulate them, expand them, then you insert the gene for this particular CAR T cells. These T cells, which are genetically engineered to express CAR cells, are expanded and then reinfused or infused into the patient. This is called autologous transplantation. And there are various ways to utilize genetic tools for the genetic engineering. They are listed here. I don't want to go into the details. Different vectors, nucleases that allow to insert this information into the cell. Just to then turn to the CAR T cells that are of greatest interest at the present time and have been most thoroughly investigated in autoimmune diseases are the CD19 CAR T cells. We know all from MS and the monoclonal antibodies that the B cell lineage going from the stem cell in the bone marrow right to the plasma cells circulating in blood are characterized by sequential display and differential display of clusters of differentiation antigens, CD19 and CD20. CD19 would also capture and deplete plasma cells and plasma blasts. Now, you insert here with this chimeric antigen receptor T cell an element that recognizes CD19 on B cells and therefore provide a very targeted possibility to identify those cells carrying CD19. I mean, with almost microsurgical precision. Once again, if you look at the procedure, I don't have to repeat this, just look at the right-hand side of this. So these CD19 positive CAR T cells would be able to recognize their antigen on these cells of the B cell lineage and cause B cell death through various mechanisms. Now, let's have a look at the lymphocytes, the B lymphocytes that are the target of these CAR T cells and how they decrease following the transfer of the CAR T cells. This is the pre-CAR T cell situation where you have these autoimmune B cells circulating. The CAR T cell brings them down, and there is, after some 100 days or so, a population with now naive B cells. So in a way, you reset a disturbed immune system characterized by the recognition sustained recognition of autoantigens. Now, another important thing is that CD19 CAR T cells allow deep tissue B-cell depletion. This was a very interesting study carried out by rheumatologists examining patients with a variety of autoimmune diseases, including inflammatory myositis, where they did a biopsy study in patients that either received the classical rituximab or CAR T cell directed to CD19. And what they found was that both groups showed B cell depletion in the periphery, but it was only CAR T cells that could generate complete B cell depletion in the lymph nodes, not rituximab. And this is shown here. This is the pre-treatment situation with a lot of B cells in the lymph nodes. This is the situation with CAR T cells. The B cells completely gone in situ, whereas quite a number are still present following rituximab therapy. So what this means, and I think it's important with where we come from with the utilization of high efficacy B-cell depleting monoclonal antibodies, it provides the hope and promise that we can achieve a durable remission by fast and deep tissue B-cell depletion. Another important aspect, of course, is that monoclonal antibodies or any antibodies cannot make it into the CNS. So their effects are executed in the periphery. CAR T cells can readily cross the blood-brain barrier and hence also reach areas of compartmentalized inflammation in the central nervous system. Now, of course, we need to be aware of the adverse events, and from the hemato-oncology field, we are aware that there are a number of toxicities occurring in a different time course. Importantly, cytokine release syndrome very early, and then ICANS or immune effector cell associated neurotoxicity, and in rare instances, for example, with a different type of CAR T cells, the BCMA directed CAR T cells, rare movement disorders, including Parkinson's. You find B-cell aplasia, hypergammaglobulinemia, and this may predispose the patients undergoing CAR T cell therapy to infections. There's also, and this is all from the oncology literature, descriptions of cases of second malignancies. This is the time course of the two most important toxicities that we are confronted with. This is the cytokine release syndrome, presenting with fever, hypertension, multiple organ failure, and later ICANS with varying degrees of encephalopathy, decreased level of consciousness, focal neurological deficits, including expressive aphasia. There may be significant cerebral edema and also seizures. And this has been correlated to the occurrence and peaks of various cytokines. And look at this, how the CAR T cells transfused into the patients now in vivo expand for a different period of time. Now, as I earlier alluded to, it was some 5 years ago or so that a group in Germany led by Georg Schett from Erlangen approached the issue, could we treat treatment refractory SLE with anti-CD19 CAR T cells, given that the antibodies are pathogenic. And yes, in a first series, they found a significant decrease in the systemic lupus disease activity score following CD19 CAR T cell therapy. And there was a follow-up, and the results were very consistent. And I can refer you to a nice review that appeared in The Lancet a couple of years ago. And this prompted the UCSF group and Stanford group to look into the possibility of utilizing such an approach for central nervous system autoimmunity. And this was initially carried out in the MOG model of EAE, of multiple sclerosis, where the infusion of CD19 CAR T cells did significantly diminish the clinical severity, but also the histological expression of the disease, with a significant diminution in the number of inflammatory infiltrates. And I've listed on the right-hand side a couple of recent reviews of CAR T cells in neurological disorders in general, and in particular in those of diseases, autoimmune diseases of the central nervous system. What are the potential advantages of CAR T cell therapy in the treatment of multiple sclerosis? Well, anti-CD19 CAR T cells, as I mentioned earlier, are already approved. CAR T cells can readily cross the blood-brain barrier and access target autoantigens in the CNS. Potent cell depletion, not only of circulating B cells, but also within peripheral tissues and the central nervous system. And a single intervention may provide long-lived efficacy. For one, it's the longevity of the CAR T product. And secondly, the idea is that you reset the disturbed immunity, B-cell immunity, and or interrupt the autoimmunity and tissue damage cycle. Now, we now have the possibility to use the 5th generation of CAR T cells, and they come with a lesser incidence and severity of significant toxicity. And these products differ in a number of molecular characteristics, different regions of the molecule. And this is a list of the CAR T cell treatments that are currently in development. For most of those, they are phase 1 trials, or phase 1/2, and one phase 2 trials. Now, the one that has been utilized so far mostly is Kyverna 101, an autologous, fully human anti-CD19 CAR T cell therapy. 2nd generation, fully human single-chain variable fragment. This is the variable fragment, and studied extensively first in B-cell lymphoma. Lower cytokine levels, hence less neurotoxicity and immunogenicity. It has been studied in a range of autoimmune indications of the central and peripheral nervous system, neuromuscular synapse, and also rheumatological disorders, vasculitides. The first report of the use of CD19 CAR T cells was recently reported by German colleagues from Hamburg in 2 patients with MS. One with relapsing, one with secondary progressive MS. And of course, it's two patients, two observations, just looking for, first of all, tolerability, and second, as a kind of suggestive hint of evidence of efficacy, at least that the T cells get in, they deplete the B cells, and apparently, with all the caveats of this very small sample, showed a stabilization of the clinical activity. So again, two patients with, sorry, I have to correct myself, one secondary and the other primary progressive MS that had been treatment refractory to ocrelizumab, underwent the usual induction therapy, lymphodepletion, conditioning regimen, then received the Kyverna CD19 CAR T cell product, and in patient 1, the acute CAR T cell expansion seen in the blood and the CSF was associated with low-grade cytokine release syndrome and a transient increase in EDSS. In the second patient, there was a more gradual CAR T cell expansion in the absence of any neurotoxicity and a stable, albeit high, EDSS. This is looking in more detail at CAR T cell expansion and B cell depletion in this patient with SPMS, where you see the expansion of the CAR T cells and the complete abrogation of B cells in blood, and similar findings in the second patient. And this is, again, sort of a hint of initial efficacy of this approach over an admittedly relatively short period of time. The colleagues in Hamburg sort of added other cases and have now a 12 months follow-up. And what I heard from Manuel Frieser, who was kind enough to lend me this slide, is that basically about the same kind of findings have been observed. Another controlled study is on its way, spearheaded by Bruce Cree from UCSF, again utilizing this Kyverna CD19 product, and another trial, phase 1, multi-center single-arm dose escalation study of a product sponsored by Bristol Myers Squibb, looks at two different cohorts, one relapsing MS, one progressive MS. They want to enroll some 36 participants. They use a classical conditioning regimen, look for 104 weeks worth of safety and tolerability data, and secondary outcomes related to efficacy, clinical, and also MRI, and in terms of immunophenotyping. Finally, there is another interesting trial utilizing autologous CAR T cells with a product that has been shown in oncology to be particularly effective and with a better safety profile. This is the AutoCell product. As I mentioned, has been shown to be very effective in leukemia and associated with relatively little toxicity. And this is the trial design. There are ascending doses of, or different doses of the cells delivered for a period of 52 weeks with an extension period to collect sustainability and safety data of this approach. The first patient was infused last October, and there are a number of patients now about to enter this trial. Now, so far we've talked about autologous cells, where the T cells are retrieved from the patient, genetically engineered, and then given back to him. Now, there are also, of course, allogeneic strategies where you have a genetically engineered T cell population that, however, comes from individuals, not the patient, and are available off the shelf, and that, of course, does offer some advantages and may overcome some of the obstacles of an autologous approach. And there is also there are efforts to look at in vivo activated or generated CAR T cells. Now, the one allogeneic CAR T cell study that has been embarked upon already is that with azer-cel, and the first patient was treated in Nebraska. And this is a product by TG Therapeutics. Now, leaving CD19 CAR T cells and focusing on CAR T cells recognizing or directed to another antigen expressed on a subpopulation of B cells, the plasma cells, plasmablasts, B-cell maturation antigen, was studied in patients with progressive multiple sclerosis. This is a recently published paper from Cell, where 5 progressive MS patients were treated with anti-BCMA CAR T-cell therapy. Treatment was well tolerated, resulted in the depletion of B cells both in the periphery and central nervous system, and also providing some impact on deactivating or deregulating microglia activity. I will finish with a brief description of what we know so far in NMOSD and MOGAD. And again, this same kind of approach, looking at the BCMA-expressing cells, was used in aquaporin antibody-positive NMOSD. Phase 1 trial interim results have been reported from China, and with this therapy, apparently the disability gauged by EDSS was diminished over time, 12 patients, and we see how this will carry on. Also from China, a very interesting study providing data from immunophenotyping, undertaking single-cell analysis of anti-BCMA CAR T-cell therapy in 5 MS patients, and it was 5 NMOSD patients, I beg your pardon. These authors found enhanced chemotaxis, allowing for a accelerated crossing of the blood-CSF barrier. Plasmablasts and plasma cells were eliminated in the CSF, and all together this suppressed neuroinflammation. Can't go into the details, but this was a very extensive, elaborate analysis. Finally, to date, to my knowledge, there's been one paper published of the clinical findings and immunological follow-up over 1 year of a patient with treatment-refractory MOGAD that received CD19-directed CAR T cells. You see here the course over time, and you can see here B cell depletion, increase in the decrease in the memory B cells, so all consistent with a beneficial impact on disturbed B cell activity in this condition. So I come to the conclusions. CAR T-cell therapies have revolutionized the treatment of hematologic malignancies. They appear to hold great promise for multiple autoimmune diseases, presumably through greater depletion of tissue-resident B cells than what can currently be achieved by antibodies. Case observations raise the possibility that this approach may work in MS, NMOSD, and MOGAD. In MS, a number of mostly phase 1 trials are underway or will soon start. CAR T-cell therapy, of course, is not without serious risks, which include short-term complications such as cytokine release syndrome and ICANS, although in autoimmune diseases like MS, NMOSD, and MOGAD, they appear to be less severe and less frequent. Longer-term complications are known, including neutropenia, hypogammaglobulinemia, and secondary malignancies. CAR T-cell therapy might offer durable remission, although it's far too early to tell. Some patients with refractory disease who were treated with CAR T-cell therapy for myasthenia gravis, SLE, and stiff person syndrome have been in remission for more than a year without treatment or with any other immune-suppressing medication. So potentially curative. And CD19 CAR T-cells appear to be safer than the BCMA CAR T-cells, which can rarely, as I mentioned, cause Parkinson's or related movement disorders. And with this, thanks to my colleagues, both in the department and abroad, I'd like to thank you so much for your attention.
Thank you, Professor Hartung, for your very excellent talk on CAR T-cell therapy in MS and other demyelinating disorders. I'm sure we can entertain a couple of questions before they break into the lunch. So may I ask a question? How common is the ICANS after the CAR T therapy? ICANS, yes. How common is it, and how to treat if you?
Well, as you saw, I think I relatively comprehensively reviewed the few data so far published in these indications, and it does occur. I think it's premature to come up with frequencies given the small sample size, but again, as was observed with SLE and myasthenia gravis and stiff person syndrome, it appears much milder, and this is probably a consequence that, in malignancies, you have a large lesion burden or cell burden, and if you attack it, you set free lots of cytokines, whereas with the CD19 or BCMA approach, there are few lesions in the central nervous system, so that it would be a simplistic explanation for the milder forms of neurotoxicity observed in the autoimmune conditions. I probably should add the following. The autologous CAR T-cell therapies have been quite labor-intensive, logistically extensive, because most of the times they are sent away, and then it takes time to produce them. So initially, the costs were something like 800,000, 900,000 euro. Now there are ways to simplify and standardize the procedures, and currently, at least in Germany and Western Europe, prices have gone down to 300,000 euro, still a lot, but of course, if you could achieve a curative, lasting response, one should consider this. And I think in its infancy is the development of the allogeneic approach, and we are eagerly awaiting the results of this one trial by TG Therapeutics.
Thank you.
Congratulations for your great lecture. Considering the concept of immunosenescence, what are your impressions about the age limit for this treatment? Whether it could be safer and more effective in the young?
Again, I think at the present time our experience is too limited, but you are making a valid point that one has to take into consideration the impact of immunosenescence. On the other hand, also inflammaging, so which may be sort of counteracting. But I think at the present time there is no. It hasn't been looked at in terms of markers of immunosenescence.
Good question. A great talk. Any thoughts on the antigen-specific CAR T cells in terms of we have all these new antibody-associated conditions and targeting only cells that are targeting that particular antigen may have less toxicity.
Right. Very good question, of course, and point, and this is being pursued with different kinds of CAR T cells, those where you combine also multiple antigen recognizing receptors. This would be the immunologist's dream in terms of the specificity. Good in situations where we know or think we know the autoantigen, MG, MOGAD, NMOSD. Quite a problem in MS where despite decades of research we haven't unequivocally identified the antigens, but there's one interesting trial about to get on from Larry Steinman's group, where they look at constructs that recognize both anoctamin, which has been implicated more recently as a potential autoantigen in MS, and an EBV peptide. You know the story about EBV and MS. So that would be a very interesting approach to deliver very targeted treatment. So all the previous attempts have just knocked out the population responsible for setting in motion, a number of pathogenic mechanisms, but not hitting the specific target.
Thank you, Professor Hartung, for that interesting discussion. And let me invite Dr. Rukshan Rupasinghe to hand over the certificate of appreciation.